Ready to go? This mic turned on. Perfect. Hello, everybody, and welcome to the Cancer Center Symposium. It's my pleasure to introduce Dr. Adam Marcus from Emory University at the Winship Cancer Center. So Adam works on metastasis, particularly focusing on LKB1 mutant lung cancer models. And he really takes an approach spanning from mice to human samples that's really quite admirable. But what I really like about what he's doing is he's really taking more of an ecological view on cancer and looking at interactions of cells and looking outside the cell. So I think you'll hear some of that today. And so he's an associate professor in the Winship Cancer Center in the Department of Hematology and Medical Oncology. And he's also the director of graduate studies and also the director of their imaging center. And finally, he's also the director of a citizen science project that I think is meant to enhance diversity and health outcomes in science. So he's really wearing quite a few hats. And I should mention he did his PhD at Penn State and then went on and did his postdoc at Emory. So he's been at Emory for, I think, most of his post-PhD career at this point. So I look forward to hearing it, and join me in welcoming Adam.
Everyone hear me all right? It's okay? Well, thanks for having me here. I appreciate the invitation. Exciting to talk today about some of our work. I think you summarized it well. We're kind of looking at how cells work together in the context of metastasis. So I'm a cell biologist by training, so we kind of use a cell biological approach to really address clinically relevant cancer questions in the laboratory. And I always kind of like to start with this quote a bit. And basically it says that, each patient's cancer may require individual specific therapy, right? And everybody knows that idea of like precision oncology and precision medicine. And ultimately it goes on to say that this could be thwarted by the emergence of genetically variant subclones or subpopulations. And ultimately it goes on to say that we need to understand this evolutionary process, right? And we think about our ideas of precision oncology and tumor evolution as being all sort of novel and intriguing, but in reality, this is by Peter Noel in 1976. So right now we're at I think a good place to actually really address some of this in terms of the technology of genomic analysis and understanding some of the evolution and mechanisms of tumor biology. And we're primarily focused in lung cancer, specifically lung adenocarcinoma. And just to make sure that we're all on the same page here, I just want to talk a little bit about some of the bigger picture concepts that we study, and one of them being the concept of intertumor heterogeneity. That is, between patients, right, and we all know this, that between patients, not everyone's tumor is alike, right? So if you take lung cancer, for example, there's a variety of different subtypes, and there's histological subtypes, and there's molecular subtypes, and this is really the whole basis of precision oncology or precision medicine. But what we also know that there's a whole other layer of complexity on top of this that exists that is more or less, ignored, at least in the treatment paradigm. We never really think about this in the idea of intra-tumor heterogeneity. And that's what's the concepts that, showed pretty simplistically with the different colors, is that, let's say, for instance, there's a blue population, there's a red population, and a yellow population. And ultimately, our treatments will kill off a certain percentage, for example, the red and the yellow, but leave the blue population, right? And it's ultimately that we know it's this blue population that grows out. And these tumor cells are able to basically evolve around all the treatments that we give it. Right? It's this sort of microevolution that happens on this really fast time scale. And until we really get at this concept of how these tumors evolve, we're still going to be left with this problem. Because that's really what cancers are. They're rapidly evolving systems. And this is something we're fascinated by. And there's a couple of really great studies that look at this. This one came out from Charles Swanton's group, who's a physician scientist in the UK. And what this is showing is basically, if you look on the top, that's representative of each patient. So like L003 is one patient, and L008 is another patient, and so forth. And what they did in this clinical study, more or less, they took multiple biopsies from the same patient. And what they're showing here is that essentially, if you biopsy multiple regions, you have different mutations, right? You have different driver mutations, even in some cases. Right? So basically this hints at, if you take this to the extreme, this hints at that each patient may actually harbor multiple diseases. And the authors of this paper end their abstract and say, this is why all our lung cancer treatments ultimately fail, because of this reason. Right? We treat, we kill part, and then the rest grows back. And we've known this for decades, right? You see patients essentially respond, they're initially sensitive, and then they relapse. So this is a challenge. It's not just in lung cancer. Probably the same data exists in numerous cancer types. And this is something that we have to deal with. And I think now we have the tools and something we're very interested in the lab. So just to give you a little bit of background on our lab, we call ourselves Tumor Escape and Metastasis Lab. It took me like 2 days to come up with the team acronym up there, and I was really proud of it. So we have that going for us. So as you mentioned, a good chunk of my lab works on KRAS LKB1. So this is a project that has existed in my lab for about 10 years. We have several grants focused on this. It really spans from the clinic. We've set up sequencing pipelines. We do very basic cell biology as well. We have GEM mice as well that basically address some of these clinically relevant mutations, and I have several people working on. We even collaborate with Drosophila geneticists. I have a grant with Melissa Gilbert-Ross on that. Another chunk of my lab is working on what I want to present today, called phenotypic heterogeneity. So looking at this intratumor heterogeneity and how different phenotypes manifest. I feel like we're missing a lot of phenotypic data. We have great genomic data. We know molecular drivers, but we don't know what biology this defines. How do these cells behave? How do they interact? And that's something that's fascinating to us. The third part of my lab, as I mentioned, is new and daunting and exciting at the same time. We developed a STEM outreach program that was just NIH funded, and we basically go to K through 12 schools throughout Georgia. We partner with the Department of Education to basically increase diversity in STEM education, and this is something that we're just getting started now and are pretty excited about it. It's called Citizen Science HD. Everything that we do, I guess even the STEM outreach part, is really related to cell biology, and we kind of use cell biology to ask clinically relevant questions. That's really what unifies a lot of what we do. I think that's where our greatest expertise is. And we're definitely, for all our studies, and this is more or less an acknowledgement, we're supported by a strong lung cancer team. Just a couple of key people on the left is Melissa Gilbert-Ross. She was a former trainee in my lab and now has a Drosophila genetics lab. There's several other people that probably some of you recognize here. Suresh Ramalingam is a clinical oncology lead for us, mainly focused on lung and so forth. So I have a whole group of people that are part of the lung cancer team that support a lot of these studies, going from pathology to genomics and so forth. And really, without this level of support, most of this would not be possible. Okay, so a little bit about the science and a little bit about the background that's of interest to us. So this is actually straight from a human lung ano, and this is like stage 3, stage 4 disease. And what you have in green, I'm just trying to figure out the best way to sort of, does that show up? There we go. Okay, so the best way to sort of, it's like impossible to navigate both. I don't know if there's a laser pointer or not. Yeah, it's hard to look up there. But anyhow, so we have what you have up there is tissue, right? And actually in the blue are multiple cells that's DAPI staining. Is there a pointer? I don't have my glasses on. Yeah, I'll see. Yep, got it. Okay, that's perfect. Thank you so much. Okay, so we have on the left, this is actually a pack of cells. So this is a typical histology seen by lung pathologists. It's actually seen in multiple solid tumor types. You know, we always think of like single cells metastasizing, but the reality is, even back to like the 1960s, people have seen these collective invasion packs. So this is like, usually it's about 20 to 30 cells that invade together in groups, and they seem to bud off the primary tumor. And these are E-cadherin positive. So I know people think EMT, they think loss of E-cadherin. These cells are heavily E-cadherin positive. That's what's in green. This is surrounding them is a collagen matrix. So there's a large amount of collagen secretion. This collagen actually ends up at the metastatic site too, so at lymph nodes and whatever the metastatic site is. And they invade in these packs. And we believe that these packs are heterotypic packs. So what I mean by that is that there are other cell types there. This is also from a patient. These are the tumor cells in here, and these are cancer-associated fibroblasts in brown surrounding them. We see this all the time, this cancer-associated fibroblast tumor cell interaction. They surround the pack. We don't know exactly what they're doing. We're very fascinated by this. We see something very similar in other tumor types as well. This is something that's a big question for us is, how do these cells talk to each other? How do they talk to the cancer-associated fibroblasts? Why do they go in packs? What's the preference of that? And that's sort of one of the underlying themes of the talk today. All right, so we try to model a lot of this in 3D. So we embed, it's either cell lines. This is an H1299 cell line. We've done patient-derived organoids, which I'll show at the end as well. And we're very interested in how these cells behave together. And we watch a lot of these videos. And when you look at it, you can start seeing these groups of cells coming off. You can see them early up there. You can see them up top here. You can see all these sorts of groups going off. And we're very fascinated by this. And we sat down, maybe like this is mainly led by a former grad student of mine, Jessica Conan. We sat down and we watched these videos over and over again. And what we started to ask was that, let me just play it again, or not. What we started to ask was the cells that come out first, these leader cells, do they have a different genome? Do they have a different behavior? Or are they just random cells that come out first and compared to the cells behind it, which we call the follower cells? So we're very fascinated by all this, and we wanted to know a little bit about how these cells behave, and if we could actually go in and actually pull out those leader cells versus the follower cells behind it. That was something that we were very fascinated by, and we said, okay, well, are these leader cells actually important? And we saw a lot of videos like this, where you have leader cells that fall off, and then the remaining pack, the remaining follower cells actually don't go anywhere. We quantified this over and over again, and basically about 86% of the time, I don't know why it doesn't want to replay, 86% of the time, let's see if that helps. Okay, 86% of the time, when the leader cell falls off, the follower cells aren't able to progress. And then we saw really cool things like this. Hopefully this one will play. We saw cool things like this, where when a leader cell comes out and it has no follower cells, it actually goes back and makes a U-turn to get the follower cells, right? So we actually see this often. You know, it really gives you the feeling, right? It gives you the feeling that it actually wants to invade as a pack. So we found this very fascinating, and we quantify all this. And I'm not going to go into the details of all the quantification, but we do a lot of image analysis sort of stuff. And basically, you know, you can see this event here when a leader cell detaches. The pack really doesn't advance anymore. The pack gets much slower. It doesn't displace and all that sort of stuff. You can also see the U-turns plotted out here. So you can see that these cells make these U-turns, and we quantify all this, and we're very fascinated by this behavior. So we asked the question, well, we felt like this was important, right? We said, okay, well, maybe these leader cells are important, but how do we get at them? And this was a platform that we developed. This should be out any day now in Nature Communications. I feel like I keep saying that for the last month, but they keep telling us any day now it should be in print. So basically, Jessica and I sat down the microscope and thought about a few different ideas on how to do this. And ultimately what we did, we tried to take the easiest approach to this, and we combined the concept of photoconversion with cell sorting. So photoconversion, it's like, if you're not familiar with these things, it's like a GFP, like a fluorescence GFP. But instead of always emitting green, you could actually and precisely pick any cell that you want and shine a UV laser on it, right? So through the microscope, like a 405 nanometer laser, and it'll precisely change that cell from green to red. So it's basically an optical highlighter, right? So if you think about reading a book and you want to highlight words in the book, this thing essentially is highlighting cells. So we did a whole bunch of validation as we can mark one cell, not the other, all that sort of stuff. We could do about 100 cells in about an hour. And we said, okay, well, let's mark the red cells. And then we put it through a cell sorter, and let's find the red cells. And we were able to do that. And we said, let's mark the follower cells, turn them red, and then pull them out. And 2 years later, this is one of those slides that's like 2 years worth of work to get it to work. We were able to do it successfully. And what you end up with are living cell populations that are highly purified based upon phenotype. So we're basically pulling out cells based upon their phenotype of, let's say, leader cellness or follower cellness, and then isolating those cell populations from a cell line, or I'll show you later, we're doing this in patient-derived organoids now. And you end up with something alive, so you could grow cell lines from it, you can grow, you could look at genomes, epigenomes. We've done a whole bunch of things that I want to sort of show you on it. And you know if it worked or not because you can actually visually see if the cell is red at the end of the day. So you have a very good criteria to know, hey, I got red cells and what percentage and all that sort of stuff. So we could use this not just for like leader and follower cell, but you could use this for anything that you could see under the microscope, right? So we want to use this for like drug-resistant cells that are invasive. We are hooking it up to hypoxia reporter next. We've done a lot of different things. So any phenotypic criteria you want, you could pull out a population of cells. So that's where I want to go with some of this now. So this is just a little bit about the setup of, we make these spheroids and they exist in like multiple dimensions. And this is the bottom half of the spheroid related to the plate, and we could image all this in 3D. And this sort of shows you how it works. So if you want a leader cell, you could zap it and turn it red. And if you want the follower cell pack, you could zap it and turn it red. We could drop a single cell in a well. We could do a whole bunch of manipulations. We're trying to get through all this. It takes time. We're trying to figure out a lot of this stuff. But what happens? So basically, when you do this, you end up, these are purified leader cells. They are highly invasive, and the first question I get is, do they maintain their invasiveness over time? So leader cells, yes. So leader cells have been in culture, in plastic, out of their 3D world for over 2 years now, and they are highly invasive. They are very mesenchymal. I'll show you some data on that. And they maintain this phenotype in plastic. Follower cells, on the other hand, are minimally invasive. I think most of this is proliferation, the expansion of the spheroid. They rarely invade. It's one of those few cases in biology where it's like literally night and day. So every single leader cell spheroid and every single follower spheroid shows this phenotype. And we end up with something very unique. Now, the thing about the follower cells, though, is that after about 2 months, they will spawn new leader cells. So there's definitely some lineage aspect to all this, is that follower cells can spawn leader cells. Leader cells only stay like leaders. So I think we're pretty fascinated by that, and we're thinking about cool lineage analysis and all that sort of stuff, but we haven't quite gotten there yet. So we've done quantification on this. This is actually for people that do image analysis. This is up on GitHub now, so people could download this 3D spheroid analysis software. But you could see, you could watch these things and you see that, the leader cell spheroids invade more, they have more branches, they have all this, different phenotypes than the follower cells. And over time, like I said, the follower cells will basically look like the parental population after a couple months. So we just did very basic stuff just to really do like some of the fun, really sort of observational things that I think are interesting. What we did was we took a pure follower cell spheroids, so those purified follower cells, and then we spike in leader cells into them. So even at like 1%, which in the initial time represents like 100 leader cells, you start seeing a trend back to chains, and then of course you reach significance at about 10% and 50%. So just by simply adding back those leader cells, you could regain the chains that are there, and you regain invasion, and these are just some examples of it. But the question is then, are those leader cells the ones that are actually leading in these newly formed chains? And what we did here is we marked the leader cells permanently red just with an RFP, and you could see that it's like 90-somewhat% of the time, the red leader cells are at the front. So the red leader cells retain leadership in this, and it's not like there's some new leader cells coming out to actually do this. So it's very fascinating to us, and we could see all these things, and there are other studies that we want to do with this. People always want to know, is there EMT and that sort of thing? And sort of the answer is sort of, kind of. We see like, and we've done this in a few cell lines now, I forgot to mention that. Follower cells are very cuboidal. You could spot them even without marking them. And it's like 90-somewhat% of these follower cells are super cuboidal. Leader cells are very mesenchymal in terms of shape. Leader cells also have a bunch of vimentin that clearly is delineated here. This is vimentin expression in leader cells here. And then the follower cells have nearly no vimentin. However, if you look at like N-cadherin, which is a marker of mesenchymalness, it's the opposite. So we don't see that change over there. This specific cell line is methylated on E-cadherin, so we couldn't actually look at that in this case. So yeah, in shape they're mesenchymal, molecular partially mesenchymal. So we were interested in following up on some of that. So we did a few other sort of mixing type experiments, and we asked if we add the leader cells to the follower cells, do the follower cells move more? And that was definitely true. But then we asked the case, what is if we just take the leader cell media? So not the leader cells themselves, but actually the media, the conditioned media that's on them for like 2 or 3 days, and we put them on the follower cells, and it's pretty fascinating. You get these follower cells now that move all over the place just by simply adding the media. So it clearly suggests that there's something in that media that seems to call the follower cells there, and you can see that they have greater displacement and greater velocity, and you can see all the cell tracks that are there. So there's something secreted going on and all that sort of stuff. So we then decided to start getting at the molecular details of this. We were like, how is this biology playing out on a molecular level? So we did these, we did this transcriptomic analysis, and there weren't as many key pathways that stood out. You do these things and you always get some hits. But the ones that really stood out were really interesting. Focal adhesion pathway, which is something that we're very familiar with. We studied that for a while. Not a huge surprise because these cells are motile. And VEGF and Notch signaling was also there, which was fascinating to us because, these aren't endothelial cells and all that sort of stuff. But, when you look at this, the first thing you think is angiogenesis, right? If you look at this, it doesn't take, a genius to say, hey, angiogenesis has a tip cell and a stalk cell, and boy, that looks a lot like a leader and a follower cell. Perhaps they're undergoing the same pathways. And that was something that we started to pursue because of the VEGF-Notch link. And we have a ton of molecular data on this, and I'm not going to get into all the details, but I'll just show you some of the key experiments. And we think that these leader-follower cells are undergoing what we call an atypical vascular mimicry. So it's not identical to angiogenesis or vasculogenesis, but it's similar. And I'll show you why. So one, the leader cells basically have a lot more secreted VEGF protein than either the followers or the parentals. If you look at specific isoforms, it's the VEGF-165 isoform that's up there. So these leader cells secrete a ton of it, which already differs from like a tip cell in angiogenesis. So in angiogenesis, it's not the tip cell that's secreting the VEGF, but in our model here, it's those leader cells that are secreting VEGF to call the follower cells, and I'll show you why we think that. If you inhibit VEGF, and we've done this siRNA, we've done inhibitors, we've done antibodies, what you end up losing the packs of cells. So you have packs here, and essentially you give anti-VEGF as well as, like I said, a whole bunch of other ways of doing this, and you start losing cellular packs. So that whole collective invasion essentially disappears with a VEGF inhibitor, suggesting that the pack is required, VEGF is required for pack formation. But there's definitely a stipulation to this, and that is VEGF is not required for the actual movement of the leader cells. So these are purified leader cells. They show no response to VEGF inhibition whatsoever. So leader cells still move. That's probably why we still get an expanding spheroid when in the previous slide. But they cannot form a pack, suggesting that the VEGF secretion is necessary to call follower cells over. So we're very fascinated by that idea. Also, a lot of the notch and DLL4 patterning that you see during angiogenesis holds up in this as well. So leader cells have DLL4 and follower cells have a ton of notch, and we've given notch inhibitors, and that also disrupts the pack, and we've done a whole bunch of knockdowns and all those things, and that's all in the paper. But essentially, it's a very similar pathway to angiogenesis in terms of pack formation. Movement, however, is related to focal adhesion kinase, as I alluded to before. The leader cells secrete a ton of fibronectin compared to the parental and follower cells. The fibronectin knockdown does inhibit leader cell motility. It completely, essentially, abrogates leader cell motility whatsoever. So if you knock down fibronectin, you get no leader cell movement whatsoever. These cells are highly dependent on it, and you could really see fibronectin imaging, what these leader cells do in terms of their secretion of fibronectin, and follower cells have virtually none. So we're very fascinated by how these fibronectin secretion pathway works. We know that it's tied to focal adhesion kinase. This is a cool assay developed by Khalid Saleda. He's in the Department of Chemistry. It was published in Nature Methods a few years back. It's like a tension sensor assay. So whenever the cells pull, it generates what's called a fret signal. So whenever these leader cells tug on the matrix, you get real-time visualization of this. So that's what all this red is here, and you can see leader cells tug a lot more than the follower cells. This is all focal adhesion kinase dependent. We could give focal adhesion kinase inhibitors to these cells, and essentially you completely disrupt movement of leader cells. So movement is a typical focal adhesion kinase fibronectin pathway, but again, pack formation is more VEGF related. So this is kind of like, where we sort of start going outside our comfort zone, and we start trying to figure out, okay, well, what does this all mean? And we have like a couple of important questions that I definitely do not have the answer to. But I think it's something that, it's going to take us years to follow up on this and years to extend this out into human lung adenocarcinoma. But I find it really fascinating is, why are there follower cells again? Why don't leader cells? They're more than happy to go, right? They move just fine. Why do either follower cells brainwash the leader cells to get them to take them with them? Or why do the leader cells, take the follower cells, right? You could think of both scenarios that exist. And then it brings up the classic question, is there an advantage to migrating in a pack, right? Why do these cells want to go in a pack? What's the advantage of all this? So some of this was in our recent publication, but most of it wasn't. We saw from day one, right when we started these things, that leader cells are hard to grow. We knew this right when we pulled them out of the Saga platform. They required a lot of nursing. These are colony formation assays. Follower cells grow great. They just take off. They have great proliferation. Leader cells struggle, and I'll show you some of that. So we did, again, very simple experiments where we mixed conditioned media. And I'll just show you this one because I think it's a cool experiment. The leader cells by themselves have very little colony number and colony area. But if you add the follower conditioned media to it, right, as well as the follower cells, so you can add those too, but if you add the media to it, you get a huge increase in the leader cell ability to grow, right? So there's something, again, there's some crosstalk, some communication that we don't know, so that's a caveat. We still don't know what it is, but we're looking at it, that seems to provide leader cell growth potential, which may perhaps be a reason why they like to take follower cells. The opposite experiment was equally cool and surprising. We took follower cells and added leader conditioned media to it, and surprisingly, this actually inhibits follower cell growth. So, in the very general, very simple interpretation that we need to look at more, there seems to be a balance here. There seems to be an equilibrium it seems that the leader cells and follower cells can each regulate their growth. And as an aside to all this, is that if we take a population of, let's say, 90% leader cells, so a huge 90% leader cells, 10% follower cells, culture them out over 3 weeks, they'll always go to 5% leader cells. So if we start with 50% leader cells, it goes to 5%. You start with 5%, it stays at 5%. There seems to be this population equilibrium that exists that, for whatever reason, this 5% leader cell growth is the most advantageous for growth. So we're working out with some population biologists at UCSD to sort of map a lot of this out and do a lot of fancy math that I won't pretend to understand. But they're working on a lot of these models and looking at basically how ecosystems grow. And they use a lot of ecological models to actually look at this. I am about 2 years from even feeling semi-confident enough to present some of that data because I'll just stumble over the math. But I think it's pretty interesting. So why don't leader cells grow well? I've studied mitosis for years, so we had to do some of this stuff. And it's a really weird mitonic phenotype. I've never seen this before. So usually we see spindle defects and all that sort of stuff. Follower cells do their 2-cell thing, which we expect. Leader cells form this really odd 4-cell stage. I don't know what to call it. But ultimately, they work themselves out back to 2 cells, and they have an enormous amount of membrane blebbing. If you looked at this, you'd probably say it's apoptosis, that they're going to die, but they don't. They have other defects as well. This project's led by a graduate student, Emily Somerville, in my lab. 43% of their defects are this what we call cytokinetic instability that I'm showing you here. They have a whole bunch of other defects. I think I have a couple movies on this too. So follower cells are happy. They divide into 2 cells. They look nice. And then the leader cells, and I just keep having issues here. The leader cells don't. So lots of blebbing, like I said, and you'll see 4 cells to 2 cells. If anyone has any ideas, I'm more than happy to I can't, for some reason, play these movies twice. But I'm very interested in what's going on the mitotic side, why these cells seem to work itself out. We've done cell cycle analysis. There's not really aneuploidy there, maybe a little bit, but not as much as you would expect from watching these videos. So right now, we're doing the in vivo experiments. I think that's obviously an important step. Right now we're doing intratracheal, we're doing subcutaneous, we're doing a whole bunch of different ways, but it's fascinating the initial results that we have. So we inject these guys in, and basically we inject parental, follower, leader. We're doing all the mixes now too to look at different ratios of these guys. But basically, follower cells grow enormous tumors, and they're more robust. It's something like 90% of mice get tumors in follower cells and about 60% in parentals. So they're super good growers. They never seem to get metastatic lymph node disease. We have not gone through all the other organs yet, so we're in the midst of doing that today. Leader cells never get primary tumors, 0. So it completely eliminates their ability to grow. However, when you actually look at the lymph nodes, and I don't have this data here, the leader cells still metastasize to the lymph nodes. So they basically don't grow primary tumors, but they just take off and leave, which is, I guess based upon the in vitro studies, what we'd expect. I think the mixes of the two would be quite fascinating for us to really look at. We're doing multiple cell lines and all that good stuff, so that's all happening, these past few months. So I want to sum up where we are, and then I kind of want to just briefly talk about where we want to go and some of the different big picture projects that we have related to this. We kind of talk about this as a symbiotic ecosystem. It's an idea that has been thrown around before. I think it's fascinating. I think the idea of treating the cancer as an ecosystem is severely understudied, and we know that it's a rapidly evolving ecosystem, and it's something that I'm new to the game, but I find it very exciting. We think that at least in this case, these leader cells require fibronectin secretion, and they require full lesion kinase. They somehow signal back through VEGF to basically call the pack. We're working out more of the molecular details on the notch side and so forth. We think that the follower cells provide some pro-survival molecule. We've done secretome analysis. we think, and we have a very interesting amount of data on this at this point, that there's a lot of, we call it metabolic heterogeneity. These leader cells, highly oxidative phosphorylation. So follower cells, all glycolysis. And we think that these follower cells are secreting lactate up to the leader cells to promote their survival, and that's something that we're really looking into at this point. So I'm working with a metabolic person, a metabolism expert at Emory to help solve some of this. So we're trying to basically understand some of these defects that occur. So when I titled it Cooperation is in Our Nature, it's literally, in our nature. We see it on all sides, and I think it'd be ignorant of us to assume that just because these are single cells and these are higher organisms, that it's not undergoing the same ecological processes. Fish, bees, right, ants, they all leave because of nutrient deprivation. That's probably why cancer cells leave too, right? Because they're deprived of nutrients. We know this forever. They typically send out sort of a search party of, bees send out search parties. You call them leaders or whatever you want to call them. They go in groups. There's a big benefit of going in a group. There's a concept called swarm intelligence. It's used in drones that basically the whole unit is much smarter than any individual organism. You could think of pack animals. You could think of things like that. I think these are totally untapped fields. You could think of Walking Dead, right? You could think of this. You could think of basically these large groups, right, are much more effective than a single zombie, right? All those sorts of ideas exist. There's a really cool organism called Dictyostelium. They are these amoeba. And I always get pretty excited about these guys. Everyone makes fun of me, but I think they're really cool. They are single-cell amoeba that are fine all by themselves. You lower their you lower the amount of nutrients, and they come together to literally form a multicellular slug in like a day. So, just a single nutrient deprivation causes them to work together. People study them in the context of altruism and how things cooperate. So, I wonder, and this kind of brings me to the last part over here, I wonder if we need to think about, these cancer cells colonizing a new island, and that we want specialists, right? If we were going to colonize an island, we don't want, 100 scientists, right, out on an island. We want people that could, build a fire maybe, build a house, right? Those sorts of things, right? So I think we need to start thinking about these models of how new islands, how new colonies develop. And, maybe this all sounds a little weird and odd, but I think it's something that is completely untapped and completely exciting. So I just want to talk a little bit about where we're going with a few of these things. I'm just going to give you really short snippets of some of our ideas. We're very interested in how leader cells promote invasion, and if cancer cells could take normal cells with them. So we work with this line called these HBEC cells. We work with these HBEC cells. These are normal bronchial epithelial cells. They are more or less normal. They're immortalized for culture. They almost never invade. This is like 24 hours, 48 hours. They just sort of hang out. However, we were kind of interested to leader cells just take everything with them. That's why we did the experiment. Leader cells are in green. You add 25% leaders, and you get these massive invasion packs, right? And you can interpret this one of two ways. You could say, well, leader cells just grab everything and go, right? It doesn't matter what it is. Or there's some other mechanism at play, and I'll show you two movies. Green leader cells embedded out, and then you have these HBEC cells, and they're not taking it out. They're calling them over. And we see this over and over again, and it makes you feel like, it gives you the feeling that, someone's brainwashing someone in this case, right? And I can make a case both ways. And I have another slide. I have another movie. Something's going on here. So I'm trying to get someone to work on this. I'm trying to get a student or a postdoc. It's an interesting idea. I don't know if anyone's ever thought about, we know about cancer-associated fibroblasts, but do normal cells end up at the metastatic site? No idea. We're very fascinated by these studies, so we're trying to get this going. I want to give a little more big picture stuff that's related more to some of the clinical work that we do. And it relates back to all our sequencing initiatives. And we've learned a ton from them, so I'm not knocking them, but I think that we're missing a whole layer of complexity on this. All of our current sequencing approaches that we use, TCGA, whatever it may be, we grind up a tissue, right? And it's a population-based mechanism. Sure, there's laser capture, but there's very little phenotype associated with that. So we're losing these rare cells. Maybe they're leader cells, maybe they're stem cells, whatever they may be. And I think we want to try a different way of actually doing this. All our sort of rare cells that probably grow out, they're just the noise, they're just the background. Maybe they're the passenger mutations that we pick up. So we're trying to use the Saga platform. We're doing this in lung, we're doing this in breast. We have lung, breast, and melanoma funding for this. We're working with the surgeons. This comes from straight downstairs up until on our microscope, and I'm having sort of issues bringing these in. Nope, that one plays. So what we want to do with our platform is say, hey, this guy looks really invasive, or this guy is drug resistant after we treated it, or this guy is, has highly proliferative properties. So we start with a phenotype, right, straight from the patient, and then we use Saga platform to actually pull out that data. So not only do we get genomic data, but we add a whole other dimension of phenotypic data, which is completely missing from most of our large-scale sequencing efforts. We think for this reason, we're essentially missing all these rare cell genomes. We're missing all these rare cell targets. And that's something that I'm heavily invested in, something that I really want to go after. And we're just sort of getting all this up and running now. Proof of principle of this that we're missing, interesting things, is that we basically did this from our cell lines. So keep in mind, it's a cell line, but you think cell lines are very similar in terms of mutation profiles, but they're not. So we took the RNA-seq data that we had, and we looked at leader cells, and we looked at follower cells, and we said, let's see if there's any changes in the genome, right? And we validated all this, and it's really striking. These leader cells have very specific mutations. A lot of them are related to metabolism. A lot of them are related to invasive biology that are completely not on anyone's radar. There are follower cell-specific mutations. This is highly reproducible. We're trying to see, are these biomarkers of leader cells and all that good stuff. We're trying to figure it out. But at the very least, maybe these mutations aren't seen in patients. Maybe they're not seen in other cell lines. I get that. But at the very least, it says that there are rare populations that are entirely missed by, like, the Cancer Cell Line Encyclopedia, all that, that are very specific to phenotypes. Whether or not these drive the biology, no clue, right? We're trying to test that now. I have an MD PhD student basically putting all these back into cells and asking, do they affect leader cell biology? So that's a project that's going on. I won't get into all the details. We looked at epigenetic analysis of leaders and followers and all that, and we collaborate with Paula Vertino, who's next door to me. She's a breast cancer epigeneticist. And basically the take-home message, so we're working on a lot of this. The take-home message is that leader cells are heavily methylated. That's shown here compared to followers and parentals. Followers and parentals. So we're very fascinated by this methylation pattern. These are all things that we want to go after. We combined this, we integrated this data set with the RNA-seq data to start looking for biomarkers. So these 4 quadrants represent, like, heavily methylated, low expression, right? Which I think is like, this sort of quadrant over here. And then we look for demethylated, high expression, and all that sort of things. And then we end up with potential biomarkers. So I have a graduate student looking at these biomarkers, seeing if we could find them in patient tissue, et cetera. So we're literally just getting started on this. And I think I just had one more thing related to pharmacology. So we did a drug screen, right? So we have a unique phenotype. So we have a phenotype, and we have unique cell lines that are, by the way, publicly available to anyone. We have these cell lines, and we said, let's find compounds that specifically kill leader cells, compounds that specifically kill follower cells. We did this with our drug screening facility. And first thing that you see is that if the leader cells and follower cells were equally sensitive globally to all drugs, they'd follow this dashed line, but they're not. It's shifted to the right. The leader cells are more resistant to almost all treatments, especially standard of care agents for lung cancer. Not surprising because they're not very proliferative, so maybe that has something to do with it. But we hit upon some really cool compounds. One of these compounds seems to specifically kill leader cells, which are in red. That's why these leader cells are rounded at the tip. We're really just getting started on this project. But I have a cool video that's actually, I've never seen anything like this. When you see these cells come out, so this is one of the videos, you end up with like these red leader cells out on the outskirts. And when we saw this, we thought they were just like dead cells, just kind of like dead, being dead out there. I don't know exactly what we were thinking. But basically, when you watch these videos, I'll show you this one. It happens kind of quick. watch this red cell come ejected out of the actual spheroid right there. So it something actually just throws it out there. And that's like a super long distance for a cell, right? So, and this is happening over just like a few hours. The follower cells are literally throwing out a dead or G1 arrested leader cell. And we're not exactly sure what's going on there, but clearly it's not happy. Maybe it's trying to grab on and keeps trying to grab onto it, and it just keeps pushing it out. Have no clue at this point. So I'll end there with just two sort of big picture overview slides. These are our big questions. I kind of alluded to them before. We're very fascinated by cooperation. We're very fascinated by getting more into the clinical world on some of these projects. We basically want to mimic what we did with our LKV1 story, get involved with the clinical people, bring some of those PDOs into our labs, which we're starting to do. We want to start thinking about targeting cooperativity. We're reaching out to a lot of the computational modelers now. We're very interested in this plasticity and lineages that exist and applying this to other imageable phenotypes. And the last sort of rant speculation is what I alluded to before. I think, and this is total speculation, but I think we're missing all the rare cancer cells. And I think that perhaps that this is why, why we're killing 95% of the tumor, and this is why there's all this relapse. And I think we need to bring more phenotype into that, which I also think maybe we need more cell biologists, right? Because we're good at basically looking at phenotype and single cell behavior and cooperation. So this is something that we definitely want to get involved in. I feel like this is something we can really contribute to, and we need partnerships. We need partnerships all over the clinic, all across multiple model organisms, as I alluded to. We need to stay focused, but I think we need to also branch out to a few different areas. And, lastly, I think just perhaps we could get at some of this, and I don't really have projects on circulating tumor cells, but maybe these clusters of cells, maybe we could actually non-invasively monitor them with circulating tumor cells. If they are indeed these cells that actually seed metastatic sites, I think that's something that provides a really cool opportunity. There was a great paper in Cell maybe a couple of years ago that showed that circulating tumor cells that go in clusters are much more efficient in seeding metastatic sites than circulating tumor cells by themselves. So that really supports the idea of cooperation, and like I said, we see all this cooperation in patients already. So I feel like this is an untapped area, and I definitely want to get involved in it. So I'll stop going on there. And basically, I just wanted to acknowledge Jessica, who was a graduate student, who's now a postdoc at MD Anderson. Sorry, this is like a 4-year-old picture of the lab. We tried updating it, but it failed 2 weeks ago. So some people didn't show up. It was a little, I forgot to tell them they weren't wearing the right clothes. So we had all sorts of issues that day. But yeah, we have a whole bunch of people at different levels of training. So I have some undergraduates and a whole variety of different people, and some of our funding. And we reach out a lot to the clinical folks and bioinformatics, pathology, and so forth to make all this happen. So I'll stop there and take questions. Yeah. Yeah. Right. So that's a good question. So it looks a little bit more epithelial-like than in the primary tumor, or at least what we see in vitro with these guys. So when they seed the metastatic site, they seem to look more epithelial-like. They also still have a ton of their matrix surrounding them. So like a metastatic lymph node will have nearly no collagen there, or we've never seen any. But when these cells get there, they still seem to either secrete their ECM or basically hijack maybe the existing cells to secrete collagen. So it seems more epithelial-like than the primary tumor would be. We're not sure what that means, right? If it sort of goes back to MET or not, but that's what we see at this point. Very early stage, yeah. Yeah, go ahead. Right. So we tried this experiment. We tried this about 6 months ago. And we said, can we get follower cells to spawn more leader cells by depriving it of it? And like all these experiments seem so easy at first, and like the devil's in the details. So we ran it, and then it's like, okay, we start dealing with mitosis and replication rates, and we couldn't convince ourselves that by basically creating environmental stress, which is what we were doing, does that like put a stress on the system, which promotes the selection of leader cells. We weren't convinced in either way. It was sort of one of these grand hypotheses, but it never really panned out the way we wanted. Perhaps we saw something. I think it's something I want to revisit. I don't think we're approaching it in the right way, but I think that idea that you're alluding to, I think that's like a really cool question, something that I want to get at. Paul?
Well, you in the beginning alluded to the fact that there's many different cells in the microenvironment.
Right. Right.
But all your experiments are just on tumor cells.
Right.
So you had myeloid, derived cells, Tregs, and you affect the leaders.
Yeah.
Going out by adding other cells in.
Yep. So we just had this conversation maybe about 4 months ago. So Ram and I are getting started on that essentially. So we're going to be doing those. What I'd like to do is sort of like pull out cells that interact with the immune cells and actually use Saga to actually pick out those cells and say, how does that interaction affect their transcriptome? And those are things that I want to use, but we have not touched any of that. Or we have not touched, like, we're just getting started, cancer-associated fibroblasts. If we add cancer-associated fibroblasts to the follower cells, they still pull them out. So I think it brings up a bigger question. Are leader cells analogous to cancer-associated fibroblasts? Are they this one and the same or not? We don't know the answer to that. But I think that whole idea of bringing in other cells is, like, absolutely on our radar. We just haven't done it yet. Yeah.
So you showed that there were specific exon mutations that fell in the leaders, not the followers.
Right.
Now, are those recurrent across leaders from the same line, or
right.
across lines?
Testing it now. So that's a great question, right? So basically, we know the number of reads that we see this in, but we're going to start dropping single leader cells into wells and asking how often we see that. And now we're just going to do the RT-PCR, and we're going to do just traditional sequencing on other lines to ask that same question. So I just don't know yet the answer to it. Absolutely. Right, and that's exactly. So I have no idea if this drives the biology of it, right? And that's a major question we have. Just, you can make stories with any genes that you see. I love seeing like actin-related genes. That makes me happy because these are invasive cells. But I have no idea. I won't pretend to say yes or not. Yeah. 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 Mm. Yeah. Right. So never tried that. I think that's a pretty cool idea to like try the gradient experiments. Yeah. There's actually a group at Tech that has these ability to monitor Georgia Tech has the ability to monitor the gradients and do that experiment. I think it's a cool idea. So integrin expression is higher in the leader cells, just based upon our array. We haven't done like protein analysis or functional analysis on that, so I don't know the answer to that specifically. But transcriptionally was upregulated in the leader cells. Yeah. Yeah. Yeah, it's a good question. I don't know the answer to it. We've done those videos because you can't obviously get that from like still shots. I just don't remember. I have to go back and look at the video. I think it's a really cool question. I don't think we ever did tracking on those leader cells in that context as we did before. Yeah, it's a really cool idea. I need to look at it. In the back there, yeah. Right. So we've never tried in the context of like inducing ECM. We have like, which I think is cool because it's kind of like a different stress. We've modulated the ECM for leader cells and modulated for follower cells to see if we could stop it, and tune it in that way. It does impact their motility, but basically they still invade. They just form networks differently. Like they may not be as chainy, like they invade in like these fence-like chains when you have leader cells by themselves. They still invade, but they don't seem to be as interconnected. But you can find like collagen densities where you can actually stop them entirely from moving because they probably need to tug on things and the traditional sort of idea. So I don't know. Yeah. All right. Awesome. Thank you.